Welcome to this scientific exploration of fermentation. Today we're going to dive deep into the physics, chemistry, thermodynamics, and materials science that transform liquid milk into delicious yogurt and cheese. Let's start with a fundamental question. What exactly is fermentation? Fermentation is a metabolic process where microorganisms, primarily lactic acid bacteria, convert carbohydrates like lactose into other compounds, releasing energy in the process. We take liquid milk, introduce specific bacterial cultures, and under controlled conditions of time and temperature, we get a completely different material with new texture, flavor, and properties. Before we understand fermentation, we need to understand our starting material. What is milk made of? Milk is approximately 87% water. It contains about 5% lactose, which is a disaccharide sugar. 3 to 4% is protein, mostly casein. 3 to 4% is fat in the form of globules, and it contains various minerals like calcium and phosphorus. Here's a visual representation of milk composition. The majority is water, with lactose being the key carbohydrate that bacteria will metabolize. Now let's zoom into the protein structure. The dominant protein in milk is casein, and it exists in a fascinating form called casein micelles. Casein micelles are colloidal particles, ranging from 50 to 500 nanometers in diameter. They're held together by calcium phosphate bridges and hydrophobic interactions. This is a simplified diagram of a casein micelle. The white spheres represent individual casein proteins, held together by calcium phosphate bridges, shown in cyan. These structures are stable at the normal pH of milk, which is around 6.6. Now let's talk about our microbial workers. What bacteria are we using? For yogurt, we commonly use Lactobacillus bulgaricus and Streptococcus thermophilus working in symbiosis. For cheese, Lactococcus lactis is often used. These are all lactic acid bacteria, meaning they produce lactic acid as their primary metabolic product. Now here's where the chemistry gets interesting. How do bacteria convert lactose into lactic acid? First, lactose, which has the formula C12H22O11, is hydrolyzed into two glucose molecules. This happens via the enzyme beta-galactosidase, also called lactase. Then, through glycolysis and homolactic fermentation, each glucose molecule is converted into two molecules of lactic acid, C3H6O3. This metabolic pathway is exergonic, meaning it releases free energy. The bacteria use this energy for growth and reproduction. But for us, the important product is the lactic acid its. Let's think about this from a thermodynamics perspective. Why does fermentation occur spontaneously? The change in Gibbs free energy, delta G, equals the change in enthalpy minus temperature times the change in entropy. For a spontaneous process, delta G must be negative. In lactose fermentation, the enthalpy change is slightly negative, meaning a small amount of heat is released. More importantly, breaking one large lactose molecule into smaller lactic acid molecules increases entropy. At typical fermentation temperatures of 30 to 45 degrees Celsius, this makes delta G negative. Here's a critical question. Why do we control temperature so carefully during fermentation? The answer lies in enzyme kinetics. Bacterial enzymes follow the Arrhenius equation, where reaction rate depends exponentially on temperature. The rate constant K equals the pre-exponential factor A times E to the negative activation energy divided by the gas constant R times temperature T. If we plot bacterial activity versus temperature, we see it increases rapidly up to an optimum, then drops sharply as enzymes denature. For yogurt bacteria, the optimum is around 42 to 45 degrees Celsius. If temperature is too low, fermentation takes too long, and other unwanted microbes might grow. If it's too hot, enzymes denature and bacteria die. This is why commercial fermentation uses precise temperature control. As lactic acid accumulates, something dramatic happens to the pH. Let's quantify this. pH is defined as the negative logarithm of hydrogen ion concentration. Fresh milk has a pH of about 6.6, slightly acidic due to dissolved carbon dioxide and phosphates. During yogurt fermentation, pH drops to around 4.0 to 4.5. This is a 100-fold increase in hydrogen ion concentration. This graph shows the pH dropping over time during fermentation. Notice the curve is steepest in the middle when bacterial activity is highest, then levels off as acids inhibit further growth. Now here's the key question that links chemistry to texture. What happens to casein micelles when pH drops? Casein has an isoelectric point at pH 4.6. This is the pH where the protein has zero net charge. At this pH, the electrostatic repulsion between micelles disappears. The calcium phosphate bridges dissolve. The casein proteins lose their colloidal stability and aggregate together. On the left, at pH 6.6, casein micelles are dispersed and stable. As acid is produced and pH drops to 4.6, shown on the right, the micelles aggregate into a gel network. This aggregation isn't random. It forms a three-dimensional gel network. This is where materials science comes in. A gel is defined as a continuous three-dimensional solid network dispersed in a liquid medium. The solid network traps the liquid, preventing flow. In yogurt, the aggregated casein proteins form the solid network. The liquid phase is whey, containing water, minerals, and whey proteins that didn't aggregate. This represents the gel structure at the microscopic level. The pink network is aggregated casein proteins. The blue represents trapped water and dissolved components. How do we measure the texture of yogurt scientifically? This is the field of rheology. Viscosity measures resistance to flow. Yogurt is a non-Newtonian fluid, meaning its viscosity changes with applied force. Viscosity eta equals shear stress tau divided by shear rate gamma dot. Yogurt exhibits shear-thinning behavior. When you stir it, the gel network partially breaks down, reducing viscosity. This is why stirred yogurt is more liquid than set yogurt. The storage modulus G' measures the elastic, solid-like behavior of the gel. Higher G' means firmer texture. This is controlled by fermentation conditions. Why does fermentation time matter for texture? Let's examine the relationship. Longer fermentation produces more lactic acid, lowering pH further below the isoelectric point. This leads to more extensive protein aggregation and a firmer gel. However, if fermentation goes too long, excessive acidification occurs. This causes syneresis, where the gel contracts and squeezes out whey. You see this as liquid separating from yogurt. For yogurt, optimal fermentation is typically 4 to 8 hours at 42 to 45 degrees Celsius. This balances acid production with gel formation. Now let's briefly discuss cheese, which uses similar principles but takes fermentation further. In cheese making, we add rennet, an enzyme that cleaves a specific peptide bond in kappa casein. This destabilizes micelles and causes rapid coagulation even before pH drops significantly. The coagulated proteins form curds, which are cut into pieces. They're heated to further expel whey, then pressed into molds. This creates a much denser protein network than yogurt. During aging, which can last months to years, residual enzymes and bacteria slowly break down proteins and fats. This proteolysis and lipolysis create the complex flavors of aged cheese. This shows the progression from fresh to aged cheese. Color change represents both chemical changes and moisture loss over time. Different cheeses use different temperature profiles during production. Why? Thermophilic bacteria like Streptococcus thermophilus and Lactobacillus helveticus thrive at 40 to 50 degrees Celsius. These are used for hard cheeses like Parmesan and Gruyere. Mesophilic bacteria like Lactococcus lactis prefer 20 to 30 degrees Celsius. These produce cheeses like cheddar and Gouda. Higher temperatures accelerate acid production, but also affect protein-protein interactions and water retention, ultimately determining cheese texture. Let's discuss flavor. It's not just about lactic acid. Some bacteria produce diacetyl, a compound that gives a buttery aroma. This comes from citrate metabolism. Acetaldehyde, produced by Lactobacillus bulgaricus, contributes to yogurt's characteristic sharp, green apple-like note. In aged cheese, lipases break down triglycerides into free fatty acids. Short-chain fatty acids, like butyric and caproic acid, create the pungent flavors of cheeses, like blue cheese. Lipase enzymes cleave ester bonds in triglycerides, producing glycerol and 3 fatty acid molecules. We mentioned calcium earlier. Let's explore its multiple roles in fermentation. At neutral pH, calcium phosphate forms colloidal bridges between casein molecules within micelles, providing stability. During acidification and gelation, calcium ions form ionic bridges between negatively charged amino acids on different protein chains, strengthening the gel network. The calcium content of milk affects final texture. Adding calcium chloride is common in cheesemaking to ensure firm curd formation, especially with pasteurized milk where some calcium is lost. Let's apply this knowledge practically. What if your yogurt doesn't set properly? If yogurt is too thin, what could be wrong? First possibility: temperature was too low, slowing bacterial activity. Second, fermentation time was too short. Third, old or inactive bacterial culture. If yogurt is too sour, the issue is likely over-fermentation. Too long a time or too high a temperature led to excessive acid production, dropping pH below optimal. If you see whey separation, this is syneresis. Causes include over-acidification, too high fermentation temperature causing protein aggregates to contract too much, or excessive agitation breaking the gel network. How does industrial fermentation differ from home fermentation? Industrial fermentation uses sophisticated equipment with temperature control accurate to within half a degree Celsius. This ensures consistent product quality, batch after batch. Milk composition is standardized, adjusting protein and fat content to exact specifications. This eliminates variability in raw materials. Pure bacterial cultures are used in precise ratios. For yogurt, a typical ratio is 1 part Lactobacillus bulgaricus to 1 part Streptococcus thermophilus. Home fermentation has more variability in temperature control and bacterial culture, but by understanding the underlying science, you can still achieve excellent results by monitoring pH and temperature carefully. Here's a fascinating detail. The two main yogurt bacteria work together in symbiosis. How? Streptococcus thermophilus grows faster initially, producing formic acid and lowering pH slightly. This creates favorable conditions for Lactobacillus bulgaricus. Lactobacillus bulgaricus has strong proteolytic activity, breaking down casein into amino acids and peptides. These compounds stimulate Streptococcus growth. This mutualistic relationship results in faster fermentation than either species alone and creates more complex flavor profiles through combined metabolite production. An important preliminary step we haven't discussed: why do we pasteurize milk before fermentation? Pasteurization at 72 degrees Celsius for 15 seconds kills pathogenic bacteria and competing microbes that would interfere with controlled fermentation. For yogurt, milk is often heated to 85 to 90 degrees Celsius. This denatures whey proteins, particularly beta-lactoglobulin, causing them to interact with casein and create a firmer texture. After heating, milk must be cooled to 42 to 45 degrees Celsius before adding bacterial culture. Adding culture to hot milk would kill the bacteria. From a thermodynamics perspective, what's the energy budget of fermentation? The free energy change for complete lactose fermentation to lactic acid is approximately negative 200 kilojoules per mole. This energy drives bacterial metabolism. However, only a small fraction of this energy is captured in bacterial biomass. Most is dissipated as heat, which is why fermentation vessels warm up slightly during the process. At the molecular level, breaking complex lactose into simpler acids increases entropy. But at the macroscopic level, we create order by forming a gel structure. This apparent paradox is resolved because energy is dissipated to the surroundings, increasing environmental entropy more than enough to compensate. Let's briefly touch on modern variations. What makes Greek yogurt different? Greek yogurt is regular yogurt that's been strained to remove much of the liquid whey. This concentrates the protein, typically doubling it to around 10%. Straining also concentrates fat if whole milk is used, and removes some lactose and water-soluble vitamins. The result is a thicker, creamier texture with higher nutritional density. Probiotic yogurts contain additional bacterial strains like Lactobacillus acidophilus or Bifidobacterium species, selected for their beneficial effects on gut health. These strains must survive the acidic environment and remain viable during shelf life. Let's consolidate what we've learned with a comprehensive summary. Lactic acid bacteria metabolize lactose through enzymatic glycolysis, producing lactic acid as the primary product. This is an exergonic process, providing energy for bacterial growth. Accumulating lactic acid lowers milk pH from approximately 6.6 to 4.0 to 4.5, creating the characteristic tang and triggering protein coagulation. When pH reaches the isoelectric point of casein at 4.6, the proteins lose their net charge and electrostatic stabilization, causing aggregation into a gel network. Temperature profoundly affects fermentation rate through its effect on enzyme kinetics, described by the Arrhenius equation. Optimal temperature balances speed with product quality. Fermentation time controls the extent of acidification, completeness of gelation, and accumulation of flavor compounds. Longer time creates firmer, more sour products, until over-fermentation causes defects. Taking a step back, what's the essence of fermentation science? Fermentation is fundamentally about engineering controlled microbial ecosystems. We select specific bacterial species, provide optimal growth conditions, and harness their metabolic activities to transform materials. We use our understanding of pH effects, protein chemistry, and colloidal science to deliberately alter material properties, converting liquid milk into solid or semi-solid products with completely different textures. Success requires balancing competing factors. Faster fermentation versus flavor development. Acid production versus texture. Shelf life versus freshness. Understanding the underlying science allows rational optimization rather than trial and error. As we conclude, consider that fermentation represents one of humanity's oldest biotechnologies, developed empirically over millennia. What our ancestors discovered through careful observation and tradition, we now understand through chemistry, microbiology, and physics. This scientific framework allows us to improve, control, and innovate while respecting traditional wisdom. We've seen how molecular-level interactions between bacteria, sugars, and proteins cascade up through multiple scales to produce the macroscopic properties we experience as texture, flavor, and nutrition. Research continues into new bacterial strains, optimized fermentation protocols, and novel applications. Understanding the fundamental science equips us to participate in this ongoing evolution of fermentation technology.